and also that the book of abstracts will be available in our website too. So now let's move on to the first presentation. We have Anna Szemik-Hojniak with the presentation Biomimetic Short Peptides in Medicine and Cosmetology.
Okay. So excuse me for delay, half a minute. Yes, I want to say that I envy my previous speakers. They had a little bit more time than I have. So first of all, good morning to all participants. And first of all, thank you very much for this golden idea to organize this free online congress and giving me chance to present our achievements. My talk is divided in effect into two parts. In general, I will give you some information on biomimetic short peptides, and in effect, those will be the peptides and oligopeptides that can be used in medicine and cosmetology, that can be potential candidates both for cosmetic products and for drugs. So because of shortage of time, I think I omit the introduction, but then I give you some results on theoretical calculations. They have been done, for two peptides, for dipeptides, and I present experimental fluorescence spectra, an assignment of theoretical structures to the issues that give this the fluorescence, and then I present also one slide on to prove that it is this sort of carrier peptide, and then I switch very briefly to three peptides, RGD analogs in mycobacterial infections, and the possibility to be used as both in cosmetic industry and in pharmacy. So all of us, we know that peptides are very interesting compounds. Peptides are short proteins in effect, and they play important roles in many biological processes like pigmentation, inflammation, cell proliferation and migration, angiogenesis, and extracellular matrix synthesis that is especially required for cosmetic products. The biocidal activity of natural peptides is effective against broad spectrum of bacterial, fungal, and viral pathogens, as well as parasites. We know here is the example of somatostatin, of peptide hormone comprised of 14 amino acids that is very important and has a great significant impact on neurotransmitters and cell proliferation. Also, I present just as an example the peptide hormone ghrelin, which is the hunger hormone, but we realized that natural peptides contained in drugs, they are isolated from the physiological secretion of animals or humans, and these processes are quite expensive. So in the time of developed biotechnological methods, in general medicine, in aesthetic medicine, and cosmetology, there is a great demand for synthetic peptides. And synthetic peptides are called biomimetics, and when they are in in the cosmetic products, we may call these products cosmeceuticals. What are the benefits of biomimetics? No, also from the definition, they mimic the action of natural peptides, but because they consist of only several amino acids that in effect can be changed, can be altered, they easily penetrate the epidermis. They regulate the action of fibroblasts, improving appearance of the skin profile. They are also excellent humidifiers, since naturally they contain a structural water, what is useful in hydration in the cosmetics for hydration therapy. Moreover, they stimulate synthesis of natural collagen and elastin by stimulating the dermis cells, leading finally to skin regeneration and application in anti-aging therapies. Moreover, they present relatively high selectivity, efficient activity, low toxicity, low molecular weight, and their synthesis is cost effective. And if it happens that they are used in drugs, there is a possibility of oral administration. In general, the biomimetics we can divide into several classes, and signal peptides are capable of stimulating skin fibroblasts. Their action leads to an increased production of collagen and elastic fibers. And on the market, there is oligopeptide 68, such a preparation. The carrier peptides are responsible for transporting and stabilizing elements such as copper and manganese. They simply complex these two metals. They carry them to the skin and allow the intake by epithelial cells. And on the market, there is the GHK-Cu3 peptides, glycine, histidyl, and lysine. And the third group, there are neurotransmitters, neurotransmitter inhibitor peptides, peptides that are capable of increasing minimal muscle activity. And first of all, they are applied in anti-aging cosmetics. So this is the summary of bioactive peptides currently marketed for inclusion as active ingredients in the skincare products. And we see here tripeptides, dipeptides, pentapeptides. We see many palmitoyl peptides, and also you see the copper mentioned here, GHK dipeptide. For organic chemists, it is not so difficult to get some sort of derivatives. And these derivatives, of course, aim to have different but still very useful properties. And
Dr. Anna, I'm sorry to interrupt. You have 1 minute, okay?
But it's impossible. So we took one heterocyclic representant quinoxaline that has many important biological properties. And we perform theoretical calculations, DFT and time-dependent DFT, and we got for such groups the quinoxaline substituted by a phenolic group and by pyridine, and dipeptide to which this scaffold was incorporated. And we followed the change of the structure, in the ground and excited state. We got this 2-dihedral angles, which were important. We derived many isomers of both of them, and we learned the spectroscopic properties and the barrier height, the barrier energy height, to have idea how they can interconvert one into another. and then to this substituted quinoxalin, the dipeptide was incorporated. And once again, we checked all these spectroscopic properties. It showed that they are different, of course. And then the excited state optimization by time, but TDDFT method allowed us to get some information how the conformation changed, and we got the theoretical values of fluorescence maxima. And when it was necessary for us to assign appropriate isomer to appropriate fluorescence band, and so it was done. And we see that, for instance, in the fluorescence spectrum, one conformer, it was assigned to the open structure and another to the closed structure. And we have proven that these peptides easily chelate to copper ions, so we can classify them as the carrier peptides. And then I switch to the antimicrobial peptides, another group. The aim was to find a new drug against tuberculosis, which is very difficult to find because the Mycobacterium tuberculosis lives and proliferates in the leukocyte cells of human bacteriophage that in effect normally kills the bacteria after the phagocytic act. So what we have done, we have synthesized 3 groups of analogs of natural peptides, and we changed the sequence of amino acids, and we have got in each group at least one. For instance, here this oligopeptide gives important 60% antiphagocytic activity, and in the analogs of cysteamine, this is the plant hormone, also this code was repeated in the synthesized peptides. And also we found one with very significant antiphagocytic activity. And then finally 9 oligopeptides were synthesized, and in some of them we found really significant antiphagocytic activity. And in the conclusion, I may say that it is the results are very interesting, because more and more bacteria are antibiotic resistant, and so those are candidates, potential drug candidates in healing tuberculosis. And in the first case, this dipeptides presented, they can be used both in cosmetic industry, and because of the presence of quinoxalin that has many interesting biochemical properties, they can be used also as potential drug candidates. Thank you very much. Thank you for your attention. And it is very difficult to present in 6 minutes, so many slides. But nevertheless, thank you very much.
You're welcome. Thank you, Anna. Now we're moving on to Celso Ferraz, contributions towards the toxicological evaluation of plant extracts and essential oils.
So, good morning. Can you hear me and see?
Yes.
Well, first of all, I would like to greet everyone and say thank you for the opportunity to present my work here. So essential oils, the essential oils market has been continuously growing for the past years, with Europe representing the biggest market share. These products are used in many industries, most of them in the food and beverages and the wellness area. and production is expected to continue growing, reaching 473 kilotons worldwide by 2027. So this demand by the industry has been accompanied with growing interest from the academia. Publications regarding essential oils and plant extracts have been continuously increasing for the past 20 years, with most of them focusing on development of new applications of these products. One thing that is noticeable is that the research in the environmental field is still very low. In fact, the number of publications regarding environmental science is only 2% of the total published papers. And although plant-derived products are generally perceived as safe and healthy, there is a question that remains mostly unanswered: how safe these products really are to the environment. Let's take a look at the most industrially produced essential oils: orange, lemon, and mint essential oils. At the moment, there is no data regarding aquatic toxicity of these products. There is some data regarding major compounds found in these essential oils. For example, limonene is usually the most abundant compound in orange and lemon essential oils, and it is reported to have toxic effects to Daphne magna at relatively low concentrations. The same for the mint essential oils. Usually the major compound is menthol, and it is also reported to have toxic effects to the fathead minnowfish. And as already said by Dr. Ana Oliveira as a little teaser in this morning presentation, the InnoVeb project aims to generate scientific data regarding essential oils and plant extracts of different plants in the Portuguese territory. That can be used to develop new products. At the moment, extracts and essential oils of 5 plants have been studied for the ecotoxicological effects, namely the rock rose, thyme, basil, immortelle, and the vastly known hop plant. So the aquatic toxicity was assessed using the freshwater crustacean Daphne magna. This is one of the recommended test organisms by regulatory agencies such as the OECD or ISO to perform aquatic toxicity tests, and all tests were performed according to the OECD guideline 202. In brief, neonates are exposed to a growing set of concentrations, and the effect, in this case was immobilization, is observed and recorded after 24 and 48 hours of exposure. Then the results are statistically analyzed, and the EC50 value is determined. Moreover, a test validation with a reference toxicant is performed, and it was done using potassium dichromate. Looking at the results, the essential oils presented different toxicities. The essential oil from immortelle showed no toxic effects up to 800 milligrams per liter. The rock rose essential oil showed some immobilization with a 48-hour EC50 of roughly 200 milligrams per liter. And by far, the most toxic essential oil was the one from thyme that presented a 48-hour EC50 of 11.20 milligrams per liter, which is low. Regarding the extracts, almost all the extracts presented no toxicity to Daphne magna, except from the chloroform extract from the flowers of hops that presented some toxic effects, although at very high concentrations. Essential oils and plant extracts can be classified for their toxicity to aquatic organisms following the globally harmonized system of classification and labeling of chemicals by the United Nations, which is also followed by the European Chemicals Agency. Of the extracts and oils that presented toxicity, only the thyme essential oil can be classified as toxic under the acute 3 category. To better understand this toxicity, and with data from our colleagues from the Plant Biotechnology Center of Beira Interior, They identified carvacrol as the most abundant compound in our essential oil, making up almost 80% of the total composition. Carvacrol is a phenolic monoterpenoid, and it is consistently reported as the major compound in essential oils obtained from this plant. And it is currently registered in the European Chemicals Agency as being toxic to aquatic organisms and aquatic life. And the high abundance of carvacrol in our essential oil can then be the explanation for the high toxicity observed. So, concluding, the essential oils from rock rose and immortelle present no risk to Daphnia magna. All the studied extracts also present no risk. The essential oil from the thyme Timbra capitata has toxic effects to Daphnia magna, and this could be explained by the high content of carvacrol present in the essential oils, and this compound is already reported to be toxic to aquatic organisms. In this case, precautions should be taken to avoid contamination of water bodies, especially by producers of these essential oils. Further toxicity assessment can be considered with other organisms to get a better understanding of the ecotoxicological effects. So finally, I have to give a special thank you to everyone involved in this product, and thank you very much.
Thank you, Celso. Now we are moving on to the next presentation, which will be given by Javier Alves, fatty acids composition, antioxidant, and antibacterial activity of ethanolic PLE extracts of 4 algae species from Galicia. Okay.
Thank you very much. Okay. Well, okay, I think this. Okay, my screen is flashing constantly. I don't understand why is this happening. Okay, is it now?
No, not yet.
Let's see.
Now we are seeing the presentation. Okay, it's okay.
You are seeing the presentation now?
Yes.
Okay. I will proceed then. I am Javier Echave from the Nutrition and Bromatology Group from the University of Vigo, and I will proceed to present one of our latest works, which involved determining the fatty acid composition of 4 common macroalgae species, as well as characterizing the bioactivities, antioxidant and antimicrobial properties of the pressurized liquid extracts of the same species. Thus, the objectives of the study were to quantify the fatty acid content of these 4 common species, as well as to determine the feasibility of pressurized liquid extractions on these species from which potential bioactivities could be exerted. These 4 species were namely Ilmantalia elongata, Ulva intestinalis, Ulva lactuca, and Phystoceira baccata, which were collected from the Ferrol Ria in Galicia, northwest of Spain. The choice of these 4 species was considered being widely distributed, especially in the European Atlantic shores. They are accounted as well for rapid and annual growth, which means that they would have a highly available biomass that could be therefore employed as a feasible source of potential alternative cosmetic ingredients. Also, it should be considered that they are, especially sea spaghetti, Ilmantalia elongata, gadwit, Ulva intestinalis, are edible and used in cuisine. Therefore, we extracted and purified the lipid content of these algae using the Fogg method. And once these purified lipids were extracted, they were derivatized to fatty acid methyl esters, which were then subjected to identification and quantification using mass spectrometry coupled with gas chromatography in the conditions, with the set of conditions that are shown. We observed that the highest fatty acid content was present in Cystoceira baccata. As it is already reported in scientific literature, microalgae are not a very good source of fatty acids. Yet, Cystoceira baccata displays relatively high concentrations of these bioactive lipids, of which are somehow remarkable. The concentrations of palmitic, myristic, palmitoleic, esteric, and oleic acids. Also, it is worth mentioning that only Cystoceira baccata seem to contain, albeit at very low concentrations, arachidonic and eicosapentaenoic acids, which are recognized as highly bioactive fatty acids. Simultaneously, we also, from the same species and the same samples, we subjected them to pressurized liquid extractions using pure ethanol as solvent. The purpose of this was to extract the greatest amount of lipophilic molecules that could be present in this species. The rest of the parameters were set up based on already optimized methods. Hence, as mentioned, we tested the antioxidant and antimicrobial activities that could display this algae. Using a DPPH radical capture assay, we determined that Ilmantalia elongata and Cystoceira baccata displayed significant capture of these radicals. Specifically, Ilmantalia elongata was able to inhibit the release of free radicals at 64 micrograms per milliliter, and Cystoceira baccata much more lower concentrations at 28 micrograms per milliliter. It is worth mentioning that for the Ulva species, we could not observe a significant inhibition of this radical liberation, which could be related to their low content in lipophilic substances that could also be argued to be due to using ethanol as solvent. For the antimicrobial assay, we employed two model species for gram-positive and gram-negative bacteria, namely Staphylococcus aureus and Escherichia coli. These were cultured in microplate at different concentrations in the range that are presented, and differences in optical density were measured by spectrophotometry. From the four species, all the four species, exert some degree of inhibition on the bacterial growth, yet only Cystoceira baccata shows significant inhibition on this same bacterial growth. Specifically, we observed an inhibition of more than 40% of inhibition for Staphylococcus aureus and more than 60% for Escherichia coli. Thus, it should be recognized that in relation to the low inhibitions shown for the previous species, this could be related to the low concentration of the extract used for the assay, as well as being related to the relatively high content in fatty acids that was accounted for Cystoceira baccata. In conclusions, from the former macroalgae species study, Cystoceira baccata accounts for the greater fatty acid levels, and Cystoceira, as well as Ilmantalia elongata, ethanolic pressurized liquid extracts, display significant antioxidant properties. As well, Cystoceira baccata was the only species to show significant antibacterial activity at these low concentrations. All these results would conclude in would conclude in assessing this species and this technique of extraction as a feasible method for including these species as potential and alternative sources of cosmetic ingredients. That will be all. Thank you all for your attention.
Thank you for your presentation. It was very interesting. And now we will listen to Paula Plasencia, bioactive properties of different extracts obtained from the aerial parts of blueberry and raspberry raw materials. Paula, are you ready?
Are you seeing my presentation?
Yes, we are seeing it.
I choose it. Okay. Well. Good morning. My name is Paula Plasencia, and I'm presenting some results of my work obtained under my doctoral program. This work is focused on upgrading the commercial potential of raspberry and blueberry plant byproducts, which are typically discarded, presenting no economic value. The outline will be sample preparation and extraction process, evaluation of extracts antioxidant activity, evaluation of extracts antimicrobial activity, and conclusions. Plant samples were obtained from a private company interested in exploring the bioresidues of raspberry and blueberry plants as sources of added value molecules. After they dried, the raspberry and blueberry aerial parts were grounded. And here we have the plants before and after being grounded. On the left, raspberry samples, on the right, blueberry samples. The extracts were obtained using ring methodologies, infusion, decoction, maceration, and ultrasound-assisted extraction, being the latter two extraction performed with an 80% ethanol water mixture. The 4 tubes on the left refer to blueberry extracts, and the 4 tubes on the right refer to raspberry extracts. The aqueous extraction process followed this diagram: decoction or infusion, filtration, and lyophilization. The hydroethanolic extraction process followed this one: maceration or ultrasound-assisted extraction, filtration, ethanol evaporation, and lyophilization. Samples extracts were named according to Table 1. Extracts were studied regarding their antioxidant properties using the oxidative hemolysis inhibition assay, and antimicrobial properties were studied through microdilution method against pathogenic bacteria. For the antioxidant evaluation, we used erythrocyte solution, a microplate reader at 690 nanometers every 10 minutes. The results were presented in IC50 at 60 and 120 minutes in microgram per milliliter, and the positive control was Trolox. For the antimicrobial evaluation, extracts were incubated, diluted, inoculated with gram-positive bacteria, Enterococcus faecalis, Listeria monocytogenes, and Staphylococcus aureus, and gram-negative bacteria, Escherichia coli, Klebsiella pneumonia, Morganella morgani, Proteus mirabilis, and Pseudomonas aeruginosa. After inoculation, they were incubated for 24 and 48 hours at 37 degrees. INT was aided, and 30 minutes more at 37 degrees, and finally our results were analyzed. Antioxidant activity results. Both raspberry and blueberry samples extracts exhibited a similar activity. with raspberry decoction and blueberry decoction extracts presenting better results with 21.9 and 20 microgram per milliliter. These extracts presented similar activity, and one of them even higher than the one of Trollocs control with 21.8 microgram per milliliter. For the antimicrobial results, all the extracts presented inhibition capacity against most of the tested bacterial strains, being the ultrasound-assisted extracts the most active ones. Raspberry samples showed the highest activity, inhibiting the gram-negative bacteria Escherichia coli, Klebsiella pneumonia, Morganella morgani, and Proteus mirabilis with a minimum inhibitory concentration of 5 milligram per milliliter. In contrast, to inhibit this bacteria, concentration of 10 milligram per milliliter of blueberry extract was needed. Regarding the gram-positive bacteria, raspberry extract presented a minimum inhibitory concentration of 2.5 milligram per milliliter against Enterococcus faecalis and methicillin-resistant Staphylococcus aureus. For blueberry extract, a minimum inhibitory concentration of 10 milligram per milliliter was determined. In conclusions, these results give us raspberry and blueberry byproducts as novel candidates for industrial application. The best extracts may be used in cosmetic formulation, upgrading those bioresidues. Next step in this work will be to test and to validate these capabilities in cosmetic formulations. Thank you for your attention, and I hope I was successful in explaining the results of the topic under discussion.
Thank you very much for your brilliant presentation. And now we will give the word to Sara Gonçalves with the evaluation of cosmetic properties of natural ingredients in the Trás-os-Montes area, a PhD project.
So I hope everybody can hear and see me.
Yes.
Okay. So I would like to thank all of you to be present here today. I would like to thank the scientific committee for choosing my work to be presented here today. It's a PhD project. It lacks results. However, they will be available along this year. It's an evaluation of cosmetic properties of natural ingredients that are common here in the Trás-os-Montes area. And as you may look upon my first page, you may start guessing which natural ingredients will be used, but more of that ahead. So firstly, we had to access what do we know about this theme, namely, what is a cosmetic? And we found a definition by the Council of the European Communities that established the laws of the member states relating to cosmetic properties, cosmetic products, that says that it's a product that's applied to the skin, to the body, with the purpose of beautifying, cleansing, or improving and enhancing attractive features. So it's everything that's a soap, a shampoo, a toothpaste, makeup, everything we use on a daily basis. And we also found out that cosmetics was used throughout the history. There are many more evidence than the ones presented here, and I'm just gonna highlight one. It's from the Middle Ages. It's Queen Elizabeth I. She used Venetian ceruse. Ceruse is a mixture of vinegar and white lead, and that would give her that white purity look that she was known for. She was quite the trendsetter back then. However, ceruse was very, very toxic to the skin. So she would apply more ceruse to cover up the damage. That would damage even more the skin, and so she would apply even more ceruse. Although in ancient times they used cosmetic with toxic properties, today it is not much more different. A study performed by the Danish Council Think Chemicals found 65 chemicals of concern in 39 products, and 3 products contain illegal ingredients in the EU, namely this mascara that is sold specific in Spain and Sweden. The natural cosmetic market has also grown. since the consumer took consciousness of the concept of natural-based ingredients. A study performed in France showed that the sales went up for natural cosmetics. 80% of the French women already have decided to buy natural or organic products, and 50% of the consumers decided to buy these products when they realized the ecological impact of a non-organic product. We also know that this is a market that is constantly growing. Analysts anticipate a global revenue to reach up to $54 million by 2027. Thus, the use of natural and organic cosmetics becomes increasingly important, and this requires a strong investigation into benefits that fruits and plants can bring to our health. So we establish 5 aims with all this knowledge that we have assessed. Firstly, we want to, after selecting the natural ingredients, we want to assess their potential to promote longevity, pro-efficacy, and antigenotoxicity. Also, the potential to promote DNA damage. The natural ingredient with the best results in these tests will be used to perform a chemical characterization and also to evaluate genoprotection in human lymphocytes. Finally, the ingredient will be evaluated if it can be incorporated in a cosmetic. So we selected 4 ingredients that are common here in Trás-os-Montes. Elderberry flower and berries. Elderberry is a wild tree. Almonds and olives that come from organic farming, and grapes that only will be harvested September this year. These last 3, almonds, olives, and grapes, come from a non-intensive agriculture. For our first aim to access if the natural ingredient has the potential to promote longevity, pro-efficacy, and antigenotoxicity, we'll be using a SMART test. SMART test uses genetic markers phenotypically observed in adult tissues, and it detects mitotic recombination. For the other test, to promote DNA damage repair, we'll be using commercial cell lines, and we'll be using the COMET assay. COMET assay can measure single and double stand breaks as also AP sites. So with all these tests, we can determine which ingredient has the best results, and that will be used to perform a phytochemical composition and also an antigenotoxic potential in human lymphocytes using also the COMET assay. Finally, for the last aim, to analyze which natural ingredient can be incorporated in a cosmetic and so benefit from its antigenotoxic properties. Studies have shown that antigenotoxic properties are related with anti-aging properties. We are currently discussing it with a corporation that is known for formulating natural and cosmetic products, and also for selling them. However, I cannot disclose any more than this. Maybe next year I have some more news for you. And I'm going to just pass right through my schedule throughout the PhD. I want to thank you all for your attention. I want to thank also Echave for funding this research, and of course, my supervisor, Professor Isabel Gaivão.
Thank you very much, Sara, for your presentation. And I wish you the best luck for the project, okay? It seems very interesting. And now we will move on to the last presentation with Silena Alves. Humulus lupulus, cosmetic application of extracts obtained from cones and vegetative parts. Silena, whenever you want.
Okay. Hi, good morning, everyone. My name is Silena Alves, and I will present my work entitled Humulus lupulus, cosmetic application of extracts obtained from cones and vegetative parts. Hop is now worldwide as an essential raw material for the beer industry, and recent studies have been revealing several plant health benefits in the beer production process. Cone of hop are used and the vegetative part, such as leaf and stem of the plant, are discarded. However, the literature indicates to have a quality interesting property. Concerning the chemical composition, hop is composed by beta acid, alpha acid, polyphenol, essential oils, and nutritional components. As I said, a hop plant has important pharmacological and biological properties, such as antioxidant, sedative, antibacterial, anti-inflammatory, estrogenic, among others. The aim of our study was study the phytochemical profile of polar extras of hop and of the essential oil of Thymus digis, and further evaluated the antibacterial and antifungal capacity. Also, a gel formulation were developed by incorporation of different concentration of hydroalcoholic ester of cones and vegetative part of the nuggets and polaris variety and of a Spendium hop sample. In addition to that, the stability of the different formulation was evaluated. For that, a hydroalcoholic extraction of cone and vegetative part of hope was carried out to phytochemical profile analysis, evaluation of antibacterial and antifungal activity, preparation of formulation contained hop extract. It was also done the obtain of essential oil from Thymus digis and the antibacterial and antifungal capacity evaluated. The determination of the phenolic profile was done by HPLC-MES. The evaluation of antimicrobial activity were performed by microdeletion growth method and the diffusion in agar test. Additionally, the stability of the gel formulation was evaluated through a series of physical chemical tests, such as pH, organoleptic analysis tester, light, relative humidity, mechanical vibration, centrifugation, and density. In our study, 30 compounds were identified, 5 and alpha and beta acid, 10 phenolic acid, and 15 flavonoid. Generally, extracts obtained by cones were the richest in phenolic compounds. Concerning of alpha and beta acid compounds, we were identified the comulpon, comulon, umulon, colupulon, adreomulon, and lupulon compounds. These compounds represent the major in cones extracts. Glycoside form of the flavonoids control, quercetin, and criosterol were identified in vegetative part of hope extras. Xanthomol, prenulated chalconoid, is present in cones as described in literature, and in the present study was found only in the nuggets variety. Flavonoids were mostly found in spontaneous hope in the vegetative part of the plant. In the preliminary stability test, the formulation showed no change in the organoleptic analysis, mechanical vibration, and in the centrifugation test, presenting acid pH with an apparent density of 1.08 to 1.14. In the accelerated stability test, the formulation showed no change in the extreme consecutive temperature variation and in the relative humidity. In the light test, the formulation showed degradation. For texture size, the consistency and the cohesive of the formulation increase comparing the gel-based formulation to the increase in the concentration of extract. In spectrophotometric test, the formulation with hydroalcoholic esters from plants cone showed greater absorption, which may be related to the chemical hindrance in alpha and beta acid. All hope extracts had no antimicrobial effect at the tested concentration. The essential oil of Thymus digis had showed antibacterial and antifungal activity against all strains of bacteria and fungi tested with rigor potential against Staphylococcus aureus, then for Pseudomonas aeruginosa. As conclusion, the sterility test did not reveal safe separation of liquation of the preparation gel sample. It's important highlighted that the present study explored the vegetative part of hop. They remain poorly studied, allowing propose them as integration in cosmetic area. In this slide, these are some references that use in our work. To finish, I would like to thank Echave and Research Center for financial support, and a special thanks for your attention.
Thank you, Silena. And now I will ask all the speakers to answer the questions that the audience has been asking. So the first question goes to Celso Ferraz. It comes from Sara Gonçalves. Celso, okay. Did you extract the essential oil yourself? If yes, which process did you use? And would there be a difference between self-extracted and commercial essential oil in terms of toxicity?
Yes, thank you, Sarah, for the question. I did not extract the essential oils myself. That was done by our colleagues in the Polytechnic of Castelo Branco. So I don't have that information on which process was used. Regarding the differences, yes, there can be a difference, especially if you collect wild plants to obtain an essential oil. The composition can vary from where the plant is collected, the time of year when it is collected. There are many environmental factors that can affect the ability of a plant to produce essential oils. Regarding commercially available, well, usually a commercially available essential oil has to have a specific composition, or at least a range of composition of the main compounds. So yes, the toxicity can vary according to the form or how the plant is collected, and also the extraction process can affect the composition of the essential oil.
Thank you, Celso. And now the next question will be to Javier from Teresa Silva Braz. Okay, regarding the pressurized liquid extraction, the ethanolic extract, did you quantify the phenolic fraction? And also, did you correlate the activities achieved with the presence or absence of some compounds?
Thank you for the question. And it's actually a very good question. We did not analyze for the moment the phenolic compounds present. Considering that using ethanol as a solvent, as many of you may be aware, it absorbs the lipophilic fraction. So this means it will absorb as well as fatty acids, also, for example, carotenoids. Which many of them, like for example, fucoxanthin or beta-carotene, are considered phenolic compounds. Thus, we aimed to correlate the exerted bioactivities and properties with the supposed level of these compounds. But we did not determine the next step to make, but no, not for the moment. I hope I'm getting a message that my internet connection is low. Well, I hope you have heard me to successfully answer your question.
Yes, Javier, in fact, it did fail, but I think it was possible to understand. Yes, thank you. And now the last question will be to Paula Plasencia from Teresa Silva Braz. And the question is, did you identify some group of compounds for the different extraction yields? And if yes, what were the differences in terms of extraction yields?
I'm sorry, can you repeat the question, please?
Yes, Paula, of course. Did you identify some group of compounds from the different extraction yields? And if yes, what were the differences in terms of extraction yields?
Okay, thank you very much for the question. We have identified some phenolic compounds by HPLC DADMES. We found some flavonols and phenolic acids, and in some cases some tannins. The yields, well, we didn't determine the yield yet because we have to decide further on work what extracts to be used, and then in that time we are going to determine also the yield of the extraction.
Okay, thank you very much, Paula. Thank you again. Hope you're enjoying the conference.